today's episode, we will be telling you more about the two types of gene editing, DNA editing and RNA editing. Let's start with, what is DNA editing?
Gene editing is a technique or methodology which attempts to modify the DNA sequence within cells, be it in cells in a dish, a culture dish, or in an animal model, and possibly in the future in the clinic, to, in most cases, enable a change of, protein expression. This could be, try to fix a broken gene, or in research purposes, just to see the function of genes or model a disease in a dish.
People usually use the example of a pair of scissors. One pair of scissors or two pairs of scissors cutting a thread and then putting something in the gap. A genetic molecular scissors is a metaphor that can be used, that you cut something, you make a change. Of course, genome editing is not always about inserting something. Sometimes it's about deleting something that's deleterious. Sometimes it's just about making a single base change. So there are many aspects to editing. It's not just cutting. Sometimes it's just changing a typo. It's like something has been spelled the wrong way, and genome editing is a way of correcting that single word and to get the spelling right.
What's the difference between DNA and RNA?
Well, the RNA could be like the letter that somebody writes. And the letter has mistakes. And the RNA editing would be a way of using a method of correcting those mistakes, I guess. RNA is downstream of DNA, and that it's a temporary molecule, unlike DNA, which is a permanent molecule.
What is RNA editing then?
Well, RNA is the signaling molecule or workhorse molecule in cells. By editing the RNA, you're not changing the underlying genome of the cell, but you can still change the phenotype of the cell, so you can change the functioning of the cell by editing the RNA. But when you do that, you're not changing the underlying DNA, so the change won't necessarily be permanent.
That sums up today's episode on gene editing. Thank you for your kind attention.